Hello dear friends, today we will study how to do the dissection of a flower for separating the different parts of the flower so that we can observe them properly. So let us first of all see what are the requirements which we need to perform this practical. First of all, we need a forceps with the help of which we can separate the different whorls of a flower. Second, of course, we need a flower, a bisexual flower. I have got a rose. It's having all the whorls intact. Then a plain sheet on which we will collect the whorls of a flower. Now, starting from this stalk-like structure, you can see this stalk-like structure of a flower. It is called the pedestal of the flower. And above this, you will find there are 5 green color leaves. These green color whorls are called the sepals. Now let us separate these sepals one by one. So I have separated the first sepal, then second, third, fourth, and the fifth one. I have separated all the sepals. You can see these are green in color. This is the first whorl of the flower. Now moving inside the flower, there is another whorl which you can see the flower. These are called the petals. These are brightly colored and are fragrant. So let us separate the second whorl of the flower. This second whorl is called the petal. So there are a number of petals present in this flower. Right? Okay. I have separated all the petals from this flower. Also, now the next important part of the flower is the stamens or the male part of the flower. You can see there are number of stamens present in this flower. Let us separate these stamens. So I will place them here. They are very minute structures. Yes. These are stamens having anther and filament. So this is the third whorl of the flower. Now finally you will see there is the swollen part of this flower. This swollen part is called the ovary, and this whole structure is above this is called the gynoecium. It is the female part of the flower. So we have separated all the whorls of a flower. First the sepals which are green in color, then petals which are fragrant and brightly colored. In this flower they are red in color. Then male parts, that is the stamens having anther and filament, and finally the ovary, gynoecium having ovary, stigma and the style. So this is how we can do the practical this dissection of the flower. I hope ये आपको समझ में आया होगा. See you in the next practical. Bye.